Our next speaker is Robert Guetig from the University. Robert is a postdoc in the group Heinz and Polinsky, and soon he will start his own lab in Germany. Robert, please.
Good morning. Thank you very much for the invitation, Jonathan. Okay, so before I start, I just want to say most of this work, essentially all of this work, was done in very close collaboration with Guetig, who is also here, in the lab at the University. And what I want to discuss today with you is basically, I just review a model that we developed in the last about 6 years about how neurons can process spatial temporal patterns of spikes. And then once we've reviewed and I've reminded you of this model, I want to apply it to 2 sensory processing tasks. One is a visual task. Here I take spike train data from the retina and see how a neuron model can decode visual features based on these spike trains. And the last part of this work, I will apply our model to speech processing. Okay, so this is almost a historical review. What started us were these 2 questions. So the first question was, can a simple neural architecture decode spike-timing-based information or not? So this, I don't know, when we started, this was a debated and really open question. And the related question was, even if the architecture would be sufficient to decode spike-timing-based information, could we come up with a simple, plausible biological learning rule that could actually realize such decoding networks? And now, obviously, you're wondering, why do we care so much about spike timing? So I just want to give you 3 reasons that were passed into this field. And the first one was that for a long time people have thought about and pointed out several computational advantages to use the actual information that is in the precise spike timing rather than just firing rates. So one argument, for example, this was put forward by Simon Thorpe, is the following. What you see is a schematic of the visual system. So here's the eye, and you project information through the visual pathway up to the prefrontal cortex. And the times that you see here is the latency with which neurons can respond to information in the visual images that's shown here. So you see, for example, here in the prefrontal cortex, you can make a decision about the presence of, let's say, an animal in the visual image in about 100 to 130 milliseconds. And this is over 7 synaptic stages. And the argument is, if you look at the firing rate in all of these networks, then there's at most 1 or 2 neurons as spikes per neuron that you can use to compute this information. Okay, so this means that unless you're averaging over large neurons that are doing the same, you just simply have to use only 1 or 2 spikes from each stage to compute what you want to know. And then, if you actually look at the way that this information here is encoded, so to do this, some people like Marcus Meister can take out the retina and put it on an electrode grid here, and then use a computer to project images onto this retina, and they record from many ganglion cells simultaneously the spiking activity. And if you look at the activity for let's say gratings here, so what you see here is the visual stimuli used in this experiment. We have essentially sinusoidal gratings here, and what varies is the spatial phase. So you see this dark bar here is moving to different spatial positions as you progress through the 8 stimuli. And if you plot the activity of 4 example cells here and different cell types, then you can see that the traditional picture or dogma that these ganglion cells encode information and send it to the brain via the number of spikes per unit time, the spike count. You can see that this variable does, for some cells at least, contain really a lot of information about the physical properties of the stimulus. So if you look at this cell here, there will be no problem to take the spike count and to get a reasonably good estimate to how this picture looked that the neuron was looking at. But then there's other neurons where this is much worse, this information. And then what Tim Golish, who did these measurements in Marcus' lab, noticed is that if you look at the first spike latency you know, if you just look here at this first spike, and you plot it as a function of the stimulus phase, you see a very nice tuning of this variable with a lot of information. We quantified it here. This is the information, the latencies versus the information in the spike count. Most cells actually have a much higher information fidelity in their latencies than in the count. So you can use this information in principle to also compute whatever you want to know about this image. So this is an example of how a temporal property of the spiking activity encodes information about a non-temporal stimulus aspect. And there's many more of these examples throughout the visual system. Basically, you can take any other sensory system and you'll find another example. This has been done over the last decade, and this has been so these results of temporal codes used for the encoding of information has been a strong motivation for us to try to understand better how neurons can actually decode this type of information. Okay, and lastly, you probably all know this work, a result about synaptic plasticity. So with the advent of patch clamp recording technology, it has become possible, roughly 10 years ago, to patch from two nearby neurons in cortex or hippocampus, and to measure the change of a synapse that's connecting these two cells with very precisely controlled spike protocols. And what people found is that if you have a caudal firing sequence, that is, you fire the presynaptic cell before the postsynaptic, you see that the synapse tends to increase strength. This is a form of ATP, it's a long-lasting change. But if you reverse the ordering such that the postsynaptic cell fires before the presynaptic cell, you get synaptic depression. And the a bit of this result is that the synapse seems to be sensitive to a very high resolution of temporal ordering, down to millisecond precision. So the synapses in the cortex or in the hippocampus seem to have the machinery to care a lot about precise spike timing. Okay, now we were very motivated, but then many people say, temporal codes are just very complicated to read out, and probably cannot do it, and we just wanted to check. So the simplest framework to check it that we came up with was to borrow from machine learning. So we took two sets of spatial temporal spike patterns and labeled them as target and null patterns. So the blue ones here are targets, let's say, and the red ones are null patterns. And now we want to test whether the neuron can decode information that's in these spike times by simply asking it to recognize target patterns and discriminate them from null patterns. Okay, so how can a neuron tell us whether this is a target or null? Before I go to our work, I just want to remind you of the perceptron. So in the perceptron, there's no time in the setup of the system, because all spike tracks are, as I show here in this example, collapsed to spike count. So you see instead of two spike times, you just have two here, indicating the firing rate. And what the perceptron does, it just computes a weighted sum of these rates, and the decision is then a thresholded version of this field. So if it's above some firing threshold, the pattern is classified as target. If it's below, it's classified as null. Okay, so this is an old model, as you all know, introduced in the 60s, that has been very powerful and successful. One of the big advantages is that it has a convergence proof. It tells us that if there is a solution, we have learning what to find. Okay, so back to the temporal problem. So now, if we go back to spatial temporal patterns of spike here, we can take a simple neuron model here. So this is the standard integral entire equation. We can solve it, and then you see that the voltage in the soma here of the cell is simply the sum of postsynaptic potentials, each weighted with the amplitude of the given synapse here. This will be our postsynaptic kernel, and the voltage is just a linear sum. So it would look like this. And now we take a similar decision rule like in the perceptron. We say if the neuron generates at least one spike in the time of the input pattern, it classifies the pattern as target. And if it remains silent, it's classified as an null. Okay, so the problem now is how do we change the synapses if we make a mistake? So here what you have is a target pattern which does not generate a spike. So I show you the subthreshold voltage trace that you see. The maximum shown here stays below the time threshold. So it's a null classification, which is an error. And the problem that we face here has been termed the temporal credit assignment problem, and essentially it means how should we credit the synapses here with this error? How much should we correct them or blame them for the neuron making a mistake here? And the solution that we proposed for this problem is to modify each synapse according to its contribution to the maximum postsynaptic voltage. So the maximum postsynaptic voltage here again is where we say this is where the error happened, this is where the neuron was closest to making the correct decision, and we penalize each synapse according to its contribution to this voltage. Note that this is fairly independent of the type of neuron model that we're using here. This is a general solution to the temporal credit assignment problem, and now given a neuron model you can compute a specific solution of this gradient. And I'm going to show you now the solution for a simple model, and later I'll discuss more complicated ones. Okay, so for the linear integrate and fire with leak, we can just compute the gradient. So the threshold is a fixed constant, so we just compute the gradient of Vmax, and what you get out is simply the postsynaptic kernel locked to the maximum voltage, and the second time that enters here is simply the time of where the synapse was active. Okay, so if we look at this graphically, so you see we have here the learning kernel. This is, as I said, the postsynaptic potential, and then each synapse gets simply blamed by the weight of this kernel. So this synapse that was very close to the maximum voltage gets a lot of change, shown here, and as we go into the past, each synapse gets less corrected. And then the synapses that were essentially too far away to make any difference at the time of the maximal voltage don't get changed at all here, and similarly synapses that fire after the maximum here, they wouldn't get any change. You can do the same for depression. A neuron fired here, but since it's a red pattern, it's a null, so it shouldn't have fired, and we can do the same. We take the postsynaptic kernel. This is essentially gradient descent on the maximum voltage, and we correct the synapses as I show here. So again, the synapses far away from the maximum in time don't get changed. Okay, so we have a learning algorithm. Let's see how it works. What we do now is we just check the storage capacity of a neuron. This was our first question. How well can it decode information that's presented in time? So we generate now random patterns. So each neuron here, each synapse in these patterns has exactly one spike. So there's no information in the time range. And we randomly label them as target and null patterns. And now what I will measure is how long the neuron takes, how many cycles to converge to a solution, given a specific number of patterns that I ask it to learn. So this number obviously will grow as I increase the number of patterns, and at some stage I will probably not have a solution, so I will, the learning time will diverge, and this is what we will call the storage capacity limit. Now also note that there's no information that the neuron can learn, because these patterns are random, so it really has to store the patterns in its synaptic weight matrix to solve this task. There's nothing to be generalized here. Yeah.
What will happen if you get
the wait. You want to learn here, for instance? I think it will do. For the LTP. So the proposal is to learn here. I don't know. I would expect that it will work. Yeah, we can discuss different forms later when I come to biological implementation, which takes a convolution of the whole voltage. But yeah. I mean, the motivation of using the maximum is simply that it's the decision variable. If it's above threshold, or that's what the classification is about. The second maximum doesn't really map to the decision, so it would be funny to use it for learning. Okay, so how do we do? So first let me show you what the perceptron will do. So in order to use the perceptron, what we have to do, I have to sort of make all the spikes here come at the same time. I want to use the same neuron model, that's why I do this trick. So by making all the spikes arriving at the same time, I make them sum. I take time out of the problem, but because the initial patterns had a spike in each afferent, the perceptron would not be able to find a solution. All the patterns would look the same. So I have to sort of sparsify and just with provision one these spikes here. This is just to make it a solvable problem for the perceptron. And what you get here is a well-known result that as you increase the number of patterns, here I plot the number of patterns divided by the number of neurons that this neuron has in its input layer. And you see that as you approach the capacity of two patterns per synapse, the learning time diverges, and that the red line here is the limit, which is known well theoretically, can be computed from statistics. Okay. So this is just to set the scale, and now the temporal algorithm. Now we go back to the patterns that are not synchronous, that have time in them, and the temporal can also realize a substantial one-to-one capacity here. It's not so crucial that this is actually higher than for the perceptron, but that it is extensive. So it scales with n as we increase the system size. Okay, so this answers the first question. Neurons seem to be very efficient, sort of plausible neural architectures to decode information in spike times, and we can apply this to many other coding schemes, not just these latency codes. And what we find is that they're actually indeed very effective. And the temporal learning algorithm that I propose here is so far has shown to be very efficient to find these solutions. Just to show you a little bit how the neuron responds. So now, if I train for, let's say, 1,000 patterns, each of them has a decision time where the neuron actually spikes. And what you see here is the histogram of all these decision times. And
you have two parameters. The parameters power and power, how these affect?
These parameters are complex. So what David is asking, how do the membrane time constant and the synaptic time constant affect the performance? Okay, but first let me talk about the decision time. So what you see here is that, so this is the length of the pattern, 0 to 500 milliseconds, and you see that after learning, the neuron almost generates a uniform distribution of decision time. Actually an important feature of this algorithm that does not specify when the neuron has to fire. Okay, so the neuron is free to select where to fire for the target patterns on its own. And this, for me, it's not a bug as some people think, but it's a feature because the neuron can select what to look for in the target pattern. Okay. If you would constrain the output time, you would strongly decrease the capacity. Now, to David's question, so there's two time constants in there, and we set the synaptic time constant always to be a quarter of the membrane time constant. That's the way we scale it here. So we have one parameter, the membrane time constant. And as you see here, this is the learning, the required number of learning cycles for two values of the learning loads, alpha equals 2 and 2.5, for different membrane time. And you see that there's optimal value here, which is in our example around 10 milliseconds. This is obviously dictated by the statistics of the limit for a particular problem. The two limits are easy to understand. So this limit here, where tau goes to 0, means that the postsynaptic potentials will stop to integrate. Okay, so at some stage you will obviously break down. There's no solution if you don't have integration, because then the classification simply is whether you have a super-threshold synapse or not. And the other extreme, in this limit, the neuron approach to the perceptron and simply integrate all the cycles. Okay, so and since you're here beyond the capacity, you also have that. So in the last part of my talk, this will be an important number, the membrane time constant, because if you have a conductance-based neuron, you can actually learn it, and the system can find this optimum by itself. Okay, now I just want to address the common criticism of this algorithm, which is how can the neuron find implement the maximum operation on the postsynapse? It's obviously something very hard to measure, that the neuron can sit there, look at its voltage over, let's say, 500 milliseconds, and then after it gets a teacher signal, go back to the synapses and say, okay, I'll correct you relative to the Vmax. Okay, so we wanted a version where we can approximate this with more plausible hardware. And what we convert to, this is not sort of something, this is just one specific example. You could have many sort of rules that would look similar. What we do here is just a convolution between the voltage and the postsynaptic kernel. That you could imagine this as some intracellular calcium. So it's just a voltage-gated convolution here of the synaptic activity. And if this value crosses some threshold, let's say the standard model of synaptic plasticity, this would be the calcium threshold for induction of synaptic plasticity, then you change the synapse by some fixed amount, and otherwise you leave it unchanged. Very simple model. Could probably be improved, but we just wanted to show in principle that the cellular hardware that's well described could realize this. And you see here the convoluted signal, and here's for comparison the voltage trace. And you see that it does exactly what you would expect, that when the voltage is high, so that this type of voltage-gated channels, for instance, would open, then this trace reaches high values, and then what I shaded here in different colors is the time where this signal would be above the plasticity induction threshold, and you see that it corresponds very nicely to the Vmax actually. Here you would also correct synapses that are suggested as a set maximum. Okay. It's a very robust rule. You don't need to tune any of this very strongly, because the system essentially tunes this itself by regulating the synaptic stress. Okay, so this is a very robust rule, and to just show you in comparison the convergence performance, this is the learning cycles for a memory load of alpha equals 1, so this means 1 pattern per synapse, and this was the temporal the analytic one. This is the correlation base. And you see, you just do roughly 1 order of magnitude slower, but otherwise you converge also to 0 error.
The flexibility in the beginning was this started 0 weight?
Yeah, so this offset is actually because we didn't fine-tune the kappa and anything. So this is just for the rule to get to the operation regime. So what Ran noticed here that initially this is very likely to be all sub-threshold, right? So you will not do any learning. So we have if this happens, there's just a constant increase of all weights, and this takes time. So yeah, half of this decrease here is already here in this initial. But the point here is simply that you can come up with a simple rule that doesn't need the global maximum and still does reasonably well. Okay, and you can look at the effective learning camera. So while you run these simulations, you can actually now you could look at the mean synaptic change. Given you have a synapse, you can look how much it changed, and then plot it relative to the Vmax on that trial, and compute the average. And you'll see that this rule, although it doesn't know anything about the maximum voltage, just approximates roughly for LTD and LTP the current that we implemented analytically in the temporal. I mean, here there's a problem here in the acosal direction. This is simply because the maximum is correlated in both directions. Okay, so let me stop here with the temporal model and go to the applications. So just summarizing, we have a biologically plausible new architecture that we have shown has a high capacity to learn a broad range of spike timing-based code. Now, because these were all artificial patterns, obviously we very much wanted to apply them to real spike trains, and I'm very happy when it became clear that you could actually use it to connect to the initial motivation. How can you process visual information with just one spike from the retina, right? Which Simon Thorpe put forward as one of the arguments for spike timing-based coding, because you simply want to be fast. So the question was, can we use this neural model now to take spike trains from the retina and do some meaningful visual processing with it? Okay. mainly to improve the processing speed, and I will show that using this model we'll also get for free contrast invariance and phase and image polarity invariance. Okay, so this is the data I showed you that we used. This is again recordings from Tim Wallish and Markus Meister. And now, taking these, we can define a number of visual tasks. So here's a simple task that we started with. We call it the luminance detection task, simply because if you imagine a region of interest, here the dashed line, and in some stimuli this is a light around this region, and in some it's dark. So what we want to discriminate is the different luminance level at this dashed line. And here you see which stimuli generated. So our null stimuli are here, 4 and 5, and the neuron has to discriminate them versus the blue one here, these two. Okay? So if we take the slide trains to these stimuli and we train the temperaton and the perceptron to discriminate between them, you see that the perceptron can solve this task very well if we give it all the spikes. Okay? Obviously, because it's using the rate code, it's doing bad or very bad if you limit the number of spikes. So this axis here is simply the number of spikes from each ganglion cell that the system is allowed to use. Okay, so for one, for instance, we take only the first spike from each ganglion cell. Okay, so the perceptron is bad here, so it doesn't make sense to try to estimate the rate from one spike. And the temperaton can solve it already with one spike with perfect performance.
So you take the threshold?
How do I get the first spike?
I'm trying to understand what you did for the perceptron.
Okay, so I say that, so the all you understand, right? The all spikes. I just take one spike. So now I can say, instead of all spikes, I just say, I take only the first 2 spikes from each ganglion cell, and if there were less, I don't give more also.
So the threshold.
Yeah.
There are more than 2.
Exactly. At most, n.
But there's a problem with the perceptron if you flip it. So instead of clipping it by the number of spikes, then 3 and 10 and 120 will be the same for n equals 2. You should clip it by the time decision.
Yeah. So I did that. It doesn't help you. I mean, that's a control. So my author is suggesting to fix the window. Yeah. So I can show you that. I have the results here. I didn't put them in this talk. Essentially, no dramatic changes. But what we also did is to look at other readout algorithms. So there's no proposed sample winner take all, which simply says that if you have an afferent population, each afferent is going to be labeled with a decision, and then the classification decision is the decision of the first afferent to find. Okay. So this you can also do. It's called sample winner take all, and you see that it does. So this would be the classic winner-take-all here. It's fairly bad. And the reason here is because it's very sensitive to noise. Okay, because it's only relying on one input synapse. Now you can, as Mauer suggested, you can improve it a little bit by saying, okay, I'm not just going to look at the first guy, but at the first 2 or 3, and this is what the n means here. And then you see if you spread it out, but this costs time, because now you're waiting again for the first 2 spikes or the first 3 spikes. You can improve it in this example. It never reaches the same performance like the perceptron. Okay, another model that was proposed, Simon Thorpe, the guy that brought this argument about the temporal constraints on visual processing, also brought a solution, which in this task is doing reasonably well. This is his model. If you want details, I can explain to you later. It's fairly complicated. And none of them are, the temporal winner-take-all and the Thorpe model are both sort of not really biologically plausible, I would say. I mean, you can come up with a story, but I think the perceptron and the temperaton are much more elegant. Yeah.
So, something to do with information with the first byte, I don't think, the first byte itself. If you just reshuffle the order of the byte, or not reshuffle, just take it from, flip it from this time after.
Mm-hmm.
Do you still have all the information from the last?
I think I didn't.
If there is nothing about temperament or about the neural code itself, if there is nothing special about the first byte agency.
I mean, the first byte agency is special in the sense that it tends to be more precise than the second and third, if that's what you mean. Yeah. So if you like say you delete the first spike from all trials, you will do much worse with it. the second spike also is not always there, right? Because the first spike is usually okay. Okay, how does it work? Let me just briefly show you the solution that the perceptron is using. Actually, embarrassingly simple. So the luminance task, what I thought here is again a schematic of the task. These are our null stimuli, the plus stimuli, and you see here that they're in stimulus space. They're nearby the two groups. So what you have to discriminate is these two stimuli versus these two, and here you see the relative latency. Okay, so this is two cells, two example cells. You see for the red stimuli, these guys fire early and these guys fire late. So this leads to a positive delta T. This is the real data histogram. For the 180-degree shift at stimuli, the target class, you see that the firing order simply reversed, and you have now negative delta. So it's very clear how you can do it with one spike, right? You just need to essentially detect the order. The simple order detection here. And there's two obvious solutions. Either the simplest is with inhibition. If inhibition comes first, like here, you don't fire because the excitation is sort of inhibited. If you reverse the order, you fire, and you can do the same with asymmetric EPSPs. If the little guy comes first, the big EPSP still reaches threshold versus if you reverse it and the little EPSP comes on the falling shoulder of the big one, you don't. So it's simple solutions. What's nice about even the simple solution is,
but the simple solution should be better than the Guetig control. So what do you do if you only have one spike? You said the problem with take all this what happens when you have noise. So this will correspond to activity in only one of these.
No, the noise there is the winner take all has other noise from Guetig on one particular trial. The guy is not the first, but the second, the one that you rely on. And this here will not hurt you. And this solution anyway, I just show for pair, and the performance here is for the whole population. So it's just to show you the simple mechanism how even with two inputs you could solve this stuff. Okay, now even though it's very simple, what it's intriguing is that if you now change the contrast of these stimuli, that's exactly what Tim did, and he records the latency tuning curves for two cells, which are shown here, for different levels of contrast. You see that there's not much distortion of the tuning curve, but there's just a global shift in the baseline latency. So if you look at the relative latencies of these two cells, they don't really feel much of the contrast change here. And this can be utilized obviously by readouts like the tempertron, which only rely on relative latencies. And you see here, for even different contrasts or mixtures of contrast, you see that the tempertron is fairly invariant in its performance, whereas the perceptron, although the absolute error is still small, feels dramatically these changes. The tempertron, even you could train it. It's not shown here, but you can train it for one level of contrast, and then the performance will be almost as good on some other contrast levels. This can generalize very nicely. Okay, so let's go to even more interesting task, which is one that perceptron cannot solve at all in one layer. So this is, we call it the polarity invariance task. Here now, what you have in the region of interest at the dashed line is either you have here an edge, which is here labeled as target, or you don't have an edge. So this is like an edge detector which shouldn't care about the contrast. This is something like a complex cell in V1 could do. And you see here that in the phase space, this looks like a hard task because you're now interleaving the two labels. So this is, as expected, this you can show also analytically. The single layer perceptron cannot do it independently of how many cells you give it if you have cosine tuned tuning. And it's like an XOR problem. And the tempertron, especially with one spike, can do it with zero error. And what you see here, and this is part of what Jonathan was, I think, addressing, that the later spikes have more noise and actually make it harder here. I'll come to this later. But now let me show you the other guy, the other readouts. So the temporal winner take all on this hard task is also much worse. It's now slightly better than the perceptron, and the Thorpe model also, they're all in the same ballpark. So now these, although they're much more sophisticated than an integrate and fire neuron, can solve this task. And again, I just want to show you the embarrassingly simple solution. So now let's look at the task here. So again, we have the red versus the blue. So they're interleaved, as I said. But now, because on the red one, either this neuron fires before this one, or 180 degrees later this one before this one, you simply have that the delta t is large, either negative or positive, whereas the target here is higher together. So you just have to detect the coincidence. And anyone would know that the coincidence detection is very easy to be implemented with the neuron. Okay. So I don't need to show you how you detect coincidence spikes. And now, if you go back to this problem here of the late spike, so what is a plausible mechanism to ignore the later spikes? And one obvious attempt is to use show some plasticity. And I could now use a model that I developed a year ago with Misha. We use it for something else. We use it actually to learn the dynamic parameters of the synapse. But in this context, I freeze all the parameters and just train the weight, but I'm using a depressing synapse here. So this is just to select, to make the first spike the one that drives the decision. Okay, you see, as you expect, as you slow down the recovery time constant, it's like you make the synapse more depressing, you recover. Even if you use the all spike condition here, you go from this value, the dash line, to perfect performance. And the values here, between 100 and 200 milliseconds is roughly what we see in the visual cortex for the recovery time. What I also vary here is the utilization time constant. This is something like the probability of release. And again, you see that for all values checked here, it does a reasonably good job. And obviously, the more depressing the synapses, the higher U, the better.
Do we really care about difference between 0.04% and 0.01%?
Yeah, some referees care about these numbers.
This is all your dynamic range.
Yeah, I mean, this is something. But no, I think it's a legitimate question if you talk about first spike latency coding. What you do with the other spikes. With such a synapse, you can easily do it. Okay. So I just want to show you.
Can you go back?
Yeah.
So without the short-term plasticity, the error of
There's a gray line here.
No, but in the bottom one, what is the scale?
Sorry.
Something's wrong.
It's here. This is a 10% error, so this is 4%.
Yeah, so the scale in the upper curve should be error of 4%.
No, error.
Not 0.04%. Not 0.04%.
Yeah, sorry. Yeah, this is fraction. I thought you were doing something else. With MATLAB. Okay, so
Enormous mistake.
So this is also a development inspired by referees. So we wanted to draw the connection now to a cell type called the complex cell. So I use this to motivate this polarity invariance. Might be interesting. So just to remind you, Hubert came up with this two cell type classes. A simple cell which is orientation selective, but also cares about state, the position of the grating. And you generate simply by summing up sort of these circular sets around receptive fields from LGN. And then you can use these cells to have a second layer to generate complex cells which are summing different simple cells to make them phase invariant. Okay, so now if you just take a toy model, 7 ganglion cells. We can sort of mimic a complex cell by training it to fire if the grating is like this, or if it's moved by phase like this. And we make it orientation selective by simply making sure that it doesn't fire if the bar here is rotated. Okay, so we can play through this, and you see that with a very simple encoding scheme. So here it's just binary. We have the dark guys fire early. These are the excitatory ones here, and the light ones fire late. Here you can simply generate a complex cell, or even a simple cell if you change the firing threshold. I don't want to go into the details. And now what you notice here, the problem is the phase invariance here was limited by the resolution of these dots. So to get a real complex cell, what I did, I just went away from the toy model, and now I sampled randomly the space here with let's say 200 cells, and I used the statistics of the latency tuning that we found in the retinal data. So the modulation was roughly 20 milliseconds in the latency response, and the baselines were also scattered with roughly 10 milliseconds difference. And then I can train a tempotron with 200 inputs on the artificial population of ganglion cells, and let's see if we get complex cell behavior. And you see, so the cell is supposed to fire only for the preferred orientations here. So it's really the color is where it fires, and it fires for all phases here very nicely. And as a prediction, what comes out of this model, and makes it very easily to test it, that you have a variation in the firing time of this complex cell, which is color coded here. So it varies. So from the red, which is late, to the blue, which is early, it varies by roughly 15 milliseconds. So this is a new sort of type of complex cell that has been proposed, which could does need 2 layers, but could work on direct LGN input and implement complex cells. So there's really a controversy, and one of the arguments against the Hubel model is that you find cells that even if you lesion the inputs to the simple cells here in LGN, the complex cells still high. So this would be one model that would address that. Okay, so I don't know how much time do I have? Okay, so now we've taken real spike trains, and we've shown that this model is sufficient to show that these can also be nicely decoded, and what we get for free with this new decoding model is that we have now an explanation for contrast invariance, polarity invariance, and phase. Okay. And this is all still very rapid processing, because it can be done using only the first spikes from the register. Okay, now this was all of spatial invariances, and it was intriguing for us to also think about invariances in the temporal domain. This is, much more complicated, because now we have a temporal readout that's supposed to deal with invariances in time. And in particular, what I'm going to talk about next is time warp invariance in sensory perception. And I think it's a fairly general problem. If you have an active cell, that's driven by some motor activity, then the time scale of the sensor will always distort you percent, right? You have this for smell or feeling, and in the auditory domain it's even worse, because you even lack the afferent motor copy, but you simply depend on the motor system of the person that you're listening to. Okay, so it has been known and described in speech for a long time. I just want to play you 2 examples. One. Okay, you hear the difference. The second utterance shown here, the spectrogram, is much slower. So although the spectral energy content, which is color-coded here, is fairly similar, you see you have this strong rise in the first formant here. The time scale seems to be simply stretched out by a factor of 2 in the second example. Actually more complicated in speech, because the time loop is dynamic. So here's an example where you see that in the beginning this word is compressed relative to here, and then the gap here is extended, so it's reversed the compression, and then again this is compressed relative to here, and it's more subtle to here. 8. But obviously you hear the difference, but I think all of you heard the word 8 in both examples. Now, why is this a problem? So the intriguing thing is that that what we do when we actually process speech, or we're looking for temporal acoustic cues, things like voice onset times or transition durations, like this would be a transition duration. So our percepts rely on these temporal cues, but they're embedded in a temporal environment which is all the time modulating by scales which are an order of magnitude larger than the actual cues. This seems to be sort of a challenge, and this was discovered. Part of this was discovered already in the late 70s with synthetic speech. So here you see two spectrograms of sort of schematics. You see here three frequencies which are the same, and the only thing that varies between the ba and the wa is the length of this transition duration. Okay. So you could vary this artificially and play to humans, and then what you get is a psychometric curve, with almost 100% bar classification goes to zero as you lengthen the transition duration. Now the problem is, or what's intriguing, that if somebody speaks slow to you, you don't hear all the B's as W's. Okay, and to mimic this scenario in this experiment, people varied the length of the vowel, and they saw that this really, if you change the speaking rate, affects the position of this category vowel. So somehow the auditory system can normalize to this perturbation. And the question is, how does it do it? And there was a lot of research on it. Most of it negative. And so what has been found is that this mechanism is cognitively impenetrable. So you can instruct the subject what you're going to do, and that you should try to prevent it. They will not be able to control it. It's really controlled by the physical speaking rate and not by the perceived speaking rate. You can play tricks and make somebody think he's listening to fast speech, and still you will get these transitions correct. It's temporally loaded. It's also not just, this was a long controversy, it's not specific to speech sound, so that it's not relying on some higher order language modules that tell you what the phonemes were, and it's already, this is related also operational and pre-articulate insights. I think by now it also has been found in monkeys. Okay, so the question is, which neural mechanisms can generate such an invariance to temporal distortion? And as I said, I think it's very important also for other senses. I just want to give you an example here of olfaction. This is data from Dima Ringberg from Guetig Farms. What you see here is the pressure in the nose cavity of the rodent, and you see, it's sniffing around. So this would be inhalation, exhalation. And now what Dima did is they give an odor, which is the dark region here, and what you see is just a raster plot of the stem, right? And you see that you don't see much, if you just align it on the stimulus. Okay, there's not much structure. But now what you can do is you can align these spikes on the onset of the inhalation. Okay, and then you get this green line here, the PSP, which is a strong response time loop to the inhalation. Okay, and then you can play this game even further. Now you can realize that the sniffing is not only distorted like speech, by one clock signal, but actually it's dynamically warped all over. Okay, and if you account for this, then so you have several points here along the sniffing cycle that you can now dynamically align, and then what you recover is the green PSTH here, where you see suddenly even the slave structure is emerging with a precision of roughly 10 milliseconds. So once you do the unwarping to the clock signal here taken from the sniffing, you suddenly get a precision in the sloppy olfactory system that's roughly comparable to vision. Well, it's actually not surprising, if you think about it. Obviously, the receptors in the nose, and this really controls how the stimulus gets to it. Right? So it's not, but still, for downstream neuron, it would be very nice if you could do this unwarping to see the structure. Okay, and that's all I'm saying. That for an active sense, I think a time warp invariant readout is always very comfortable to not have to worry about this. Okay, so what's the general solution? So obviously you need to adjust your perceptual clock in the neuron to compensate for the time scale fluctuation. Now in olfaction this might be easier to do because you have the motor, you're doing your own sniffing, so you have the clock in principle. In audition, this is much harder because you don't know how fast the other people are speaking. So in audition, at least, you have to somehow infer the speaking rate from the input itself. Okay. So what is the clock in a neuron? So in the initial model that I showed you, we had the time constant here that David asked about, which is just controlling the leak of the membrane, and this was based on current, or using current-based synapses, where each synapse injected a current into this membrane. Now, a real synapse is doing something else. A real synapse is actually opening holes in the membrane and changing the conductance. Okay, now, if you incorporate sort of the simplest version of these conductance-based synapses in the model of this flavor, what you get is that the leak conductance, this leak conductance here 1, is actually the leak term in the voltage. It's the leak plus the synaptic conductance. Okay, and this means that the total synaptic conductance, this is the sum of all conductances, so excitatory and inhibitory, controls the effective clock of the cell. Okay, so the effective time constant is the resting time constant divided by the total conductance. And if you now go to a regime where the synaptic conductance is large relative to the leak, then you have the interesting observation that the clock scales inversely to the conductance. And this is exactly what we want. So if you realize that, so if you would scale time by a factor of 2, let's say, then the rate of the spike, goes by 1/2. Because we stretch time by 2, so only half the inputs arrive. So the conductance also goes down to 1/2. And then because it was in the denominator, the membrane time constant, scales like 2. Okay, so the clock of the neuron just automatically adjusts to the time scale of the input if we're in this high conductance regime. Okay, so.
But also the amplitude of the speech change to the fine rate of the neuron.
You mean the volume?
Volume.
Yeah.
Then change the clock.
Yeah, so I can discuss it a little bit in the when I talk about the front end. So what we're assuming that the conversion of the speech in the periphery to spikes that we're going to use is such that you somehow encode some elementary acoustic events, just that don't change in number but just in time. That sort of track.
Is it supported by the data?
Well, it depends who you ask, but I mean, essentially what we're using is some energy threshold crossing in the spectrogram. I think it's not wild, but obviously I don't have a I don't have a mechanistic model of how this is done and where it's done.
Actually, both amplitude and events in time are related to information processing. This is really well done as time.
Yeah. Okay, so is it feasible biologically? I mean, questions like this. I will not go into volume tuning, but I will just sort of talk about, assuming we have a fixed volume. I'm actually using real speech, so the modulations that you have in there are taken care of. I don't have screaming people in the database. But, okay, we need to generalize the learning rules, conductances, and then can we use it for speech? Okay, so what I mentioned before is that our principle to use Vmax was actually general and independent of the neuron model. This I could use for the dynamic synapses with Misha-Solik, and now we can also use it for the conductances. As long as we can compute this derivative, we can derive the temperature learning. Okay, what I want to show you here is simply what we expect, what we mean by time open variance. So now we have a spike train here and generates a voltage trace. And now if we stretch the times of the spikes by a factor of 2, we get a new voltage trace, and if we compress, we also get a voltage trace. And now if I rescale each voltage trace here on the time axis and overlay them, what you see here is an example of not time open variance, okay? Because this was just a tone model. We're not in the high conductance regime, so you see that the voltages are very different. Now, if we do the same in a neuron where we can go to the high conductance regime simply by having more inputs, like here. So here we have now 500 synapses, and I do the same. I stretch by a factor of 2, compute the voltage, and then rescale them to compare them. You see that they're very, very similar. Okay, so here the automatic time unwarping mechanism, almost works perfectly. So we can now go to the storage capacity task and see if it works with time warp, just to give us an idea. And you see here that it, for the parameters we choose, which is 100 milliseconds resting membrane time constant and synaptic time constant 1 millisecond, we get really the invariance up to a factor of twofold compression or dilation of the time, which is roughly the physiological range of speech which we want to be in. If you go beyond, it breaks down. So we can actually, we can also, this was for global time warp, we can also do it for dynamic time warp. You see that this is the current based, for comparison, the current based neuron. It gets even worse for dynamic time warp. Here it doesn't matter as we expect because it's local, the correction. Now, one important parameter, as I mentioned, was that we're in the high conductance regime. So in the previous equation I had the 1 plus g, so the 1 should be small relative to g, and this is can be expressed as the resting membrane time constant. The larger it is, the better the performance, you see here. This is for this point. And similarly, the smaller the synaptic time constant, this one is uncorrected, so this will eventually also cause interference, and the smaller you make it, the less it matters. This just to show that they are both graceful, there's no catastrophic collapse if you change them. Okay, now after training we can actually look at the voltage traces, and this is for a trained pattern, and you see very strongly here the difference. This is the conductance-based neuron, which simply doesn't see the time warping that was used here in color code to generate the different voltage traces. Okay, so this was time of 0.4-fold compression to 2.5 dilation. What's interesting for the speech application is that once you've learned a particular statistic, you can now take a completely new pattern, which is shown here. The neuron has never seen this before, and still it's time warp invariant, because the conductance-based mechanism is automatic. It works for given statistics. And this would explain, for instance, why the pre-articulate infant shows this effect for phonics, even though it has never learned this phonics. And it's also a big advantage against the state-of-the-art machines that use templates and dynamical linear programming to unwarp speech examples, but they always need a template. This doesn't have any template. Let me skip this. So let's go to the speech application. So now we need to get from sound to spikes to use this model. So what we do is we compute the spectrogram. This is something that for you the cochlear does usually. We smooth it in time and compute some psychophysically motivated scales. So this is called the Mayer scale. It's a logarithmic scale in the frequency range. There's a temporal smoothing stage, and then we just threshold the resulting frequency bands by thresholds for different energy levels. For here we have on and offset neurons, and each threshold crossing generates a spike. So just show you an example. So here would be a sound of 1. This is the spectrogram. Here you see this typical 4-month rising. Now I just show you now examples of these 3 different frequency bands. We have 31 of them, or 32 maybe. This is in time now. This is normalized to 1, to the maximum. And now we put the thresholds. So each crossing will now here give a spike. The maximum gives a spike, and the drop. The maximum is obviously also physiologically problematic. No, because you need to normalize somehow globally to the maximum. Okay, this is how our spikes look once we run it through this front end. So we see that the spike can essentially trace out the spectrogram. So here each color codes the level of the threshold. The circles are onset and the squares are offset. Okay, now I go to one of these meaningless databases from computer science. This is the TA46 digit task. It's isolated words from 0 to 9. It's like the handwritten digits, but for sound. It's clear speech, 16 speakers, female and male, and we have a fixed training and test set. So we can compare to other people. This is our result. So now we train 20 integrated sine neurons. So we have 2 neurons for each digit, one for the male and one for the female. And this is the performance we get for the, what we call biological parameters. You see that it's very good. There's a few problems here with 5, although it's still far below 1% error. This is a well-known hard digit. There are some problems with the 2. Just to give you the idea of the scale, so we get an overall word error of 0.0017, and this is very comparable to state-of-the-art machines like the Swings 4, the same error, and the HDK. So this is Carnegie Mellon University and Cambridge University. This is hidden Markov models that were developed over the last 2 decades.
What is the fundamental difference? What are they doing better or worse?
They don't have a built-in time warp invariant. So this is hidden Markov models that it's a completely different architecture. It's a probabilistic model.
Yeah, I know what it is. But just these numbers don't really tell you in what sense you're better or worse.
Yeah, I mean, this is numbers that are published in the speech community to show you how good they are. So I just did the same number.
You want a test where there's a real difference.
Yeah, obviously, I mean the isolated, single, small vocabulary. That this is not, where the frontier runs today. You want connected speech, large vocabularies, and noise. And obviously, this is things we're working on our plan. But I just want to show off here. If we, if I relax the constraints on these parameters, we can. But as you know, they're meaningless numbers. Yeah. So it's just we can do this. We can do not so bad. Okay. I just show you what we have now. We have neurons that can do this detection. So we can actually, because they're in our computer, we can look at their parameters and weights. And so this is one example of the male 1 detector. And it's just just to get some idea how it operates. So you see here for each of the thresholds. So this is the onset and the offset threshold. And for each frequency, you have one afferent, and this is its weight. So the warm colors are excitatory, and the blue ones are inhibitory. And what you see is how the neuron uses both excitation and inhibition to go to the high conductance range. And then this diagonal band here corresponds to this formant, which is the characteristic of the word 1, which is detected. Down here you see the average voltages. The blue is for the target, and the red is for the null pattern. And you see that there's a very wide margin, which makes the system very robust.
Yeah. Is it a common practice to separate males and females in auditory explanation?
Yeah, even also boys and girls. You would usually you have 4 classes. Yeah. But I mean, once you train, the system doesn't need to know, right? It's just you just have these detectors, but it's not labeled afterwards.
I thought you had a detector for male 4, female 4.
It's true, but when you do the recognition, you don't tell it whether it's a male. You just look at which detector detects, but they're not. Okay, so there you can, this is just an example of the male 4. And what I want to show you here that what I said in the beginning, that the system chooses where to make the decision. Here it was in the beginning of the word, and here at the end. So the liberty of the temporal algorithm to choose time, I think it's an essential part of allowing it to find the right solution. You don't need to tell it how to solve this. Okay, so now we can also increase the database. We can go to more speakers. This is just to show how the same solution that was derived from one database transfers to some other database, and now without retraining, this is new dialects and new speakers, you get a much bigger error, obviously 10%, and if we do the same for the state-of-the-art machines, they even worse. Okay, so this is just to show that even in that dimension, we're not so bad.
Are these models comparable in terms of complexity, number of parameters, training sets?
No, I think these are much more complex.
Did you try to quantify it? Yeah. Besides the fact that it may be more biologically plausible.
No, but no, no, I understand.
Especially as a model.
Yeah. Actually, I'm not so interested. I mean, okay, I'll show you these numbers because we're excited about them, but they're not the fundamental part of our work. I mean, we were interested in the biological mechanisms to do time warp invariant, pattern recognition. I mean, I would be happy if other people do this, to really look why the hidden Markov models in these examples are doing worse, but it's not.
No, but just compare give models and it says
Yeah, I don't know how to compute it, but
you have some capacity.
yeah.
When you calculate
I think in terms of parameters, these are much bigger class. Okay, just to show you that it's also not specific to humans, I just applied it to some birdsong detection. So this is data from Richard Hanloser, who they have actually the problem in the lab that they need to detect the syllables of these zebra finches. So he just for fun he sent me some of his songs which were labeled into training segments like this. So you have here 7 syllables I think that they want to detect, and we just wanted to see whether how robust our front end, the whole algorithm is to different species. So, and this is the performance. You see that it does very, very well. So, there's 3 errors, which I think are labeling errors, and not. If you look at them. Okay, but it's just a point here. There's nothing much fine-tuned about this technique. You can basically completely different auditory signal, and it does the same job very well. Okay, so what are some predictions? So plasticity should be shunting sensitive. So I think this is actually, most people would agree that if you shunt the postsynaptic compartment and you prevent some calcium channels that are voltage gated to open, you will interfere with plasticity. So this is probably not very drastic prediction. Then we would say that auditory neurons that are dedicated to temporal speech cues should have very small Lie conductances to allow for the control of the time scale by the synaptic conductances. Then this is something maybe of clinical relevance, that if you disrupt the balance between excitation and inhibition so that you don't allow the system to go to this high conductance state by balancing the two, you should interfere with time of invariant speech perception. And discussing with Mike Merzenich, this is something that he was very interested in. Because that's their theory, what you see in the aging mammalian auditory system. And then, I don't have time to go into this much, but there's a broad class of time perception illusions that we think are at least consistent with this conductance-based mechanism of modulating the time at which the system operates. Okay, finally, I just want to thank Chaim and Eddie, who helped a lot with this work. Markus Meister and Tim Golish for the data on the Retinal, and Michel Sodek's group I worked with for a year now to develop the STP version of this. If anyone is interested to work in this beautiful building here, they're located in Göttingen, you're welcome to stop.
Questions?
The learning of data happens in real time. You don't have a delay for the system to adapt for the speed of the signal.
Which part you're talking about, the speed?
Yes.
So, the better the time warp?
Yes.
So, the system is trained with a natural time warp. So we have, natural speech. So there's a range of time warps for which the system has to learn to not be sensitive to. So I think if you would take now an example that's outside this range of what was in your training set, you would not be able to generalize. I don't know.
So the system does not adapt in real time to the actual.
No. Within the range of the training set, it does. I mean, the robustness. You saw that beyond beta equals to 2, like in the synthetic model, the invariance breaks down. I think this is also because we didn't train in this range.
But you have to be trained in all the different rates of speech. You have to see examples.
So the way the mechanism works is that once you get to the high conductance regime, if the parameters are right, in theory you would be time of invariant training in that class of system. It's simply because your effective time constant of the neuron rescales. So you don't need to learn every time warp. You just need to get to the right regime, And this will be limited by the range of time warp that you see in your experience usually.
Roughly the time warp is the way in this model can not scalar scaling of the time sequence, but maybe like a speed up.
You mean dynamic time warp?
Yeah.
Yeah. So this we showed. I mean, part of the so this is part of the strength that is sort of confined to the scale of the resting membrane time warp. So 100 milliseconds. That's the range in which the time warp is correct. If you change the time warp beyond this time scale, which is typical in speech, it will readjust.
What about the operation of the speed energy?
That's obviously moving that direction. Yeah, if you have a more full morphology, for instance. So from a practical point of view, I try to work with models that have an exact solution simply because of the simulation. Now, if you're courageous enough and go to models that you integrate numerically, which is like 3 orders of magnitude in runtime that you pay for it, then I think there's many interesting questions. I mean, if you have full dendrite, you could imagine that you have many effective time scales distributed over the dendrite. Things like this. Yeah, but also, the more complicated the model, the better you need to specify the context. Like you would have to know where input to write, and before we get this type of information, there's not much point.
Regarding the time warp, we also predict that there are no rest tracks. They might know the scaling without achieving.
Yeah, it's interesting. I haven't thought of it. I mean, yeah, it depends how much you allow them to learn, right? We can put them and see what the system chooses for them to do. So any nonlinearity, any also non-synaptic voltage gated currents or to be tested, they don't help you. But the main point is that the time constants usually are beyond what is interfering here. So the time warp invariants operate on the time scale. So the conductance is delivered on the time scale of tau s, which is very small. And after that, the shunting is done. And I don't know of these non-synaptic conductances that operate on this time. But like H currents, I typically have 50 minutes. I think that's why the effects are very weak on interference.
Thank you very much.
Yeah, but here's the transient conductance, the shunting. The fact that the leak, sort of, whenever the conductance is open, the synaptic one, this is a short event, and if it comes, so let's say you have 2 events and you now restore time, right? So if the distortion is small relative to tau s where the conductance is delivered, something on the long range will not hurt you. No, we checked it.
Any more questions? So let's thank our speaker.